So today's lecture, I'm going to finish up the slides that we didn't get to last time. So we'll start with that, and then we'll get into what we are going to talk about today. So last time, we essentially left off with the growth cycle and talking about the different stages of growth and talking about growth in batch versus continuous culture. So now we're talking about a little bit about what happens when nutrients are either high or low, and what do cells do in those cases. So in the first case we want to talk about are biofilms. So essentially biofilms are communities of bacteria that attach to different surfaces, and then they can grow there. So they're trying to stick to a surface because essentially there is lots of nutrients. So it's a situation that occurs when things are in high nutrients. So they want to try as best as they can to stay there and just take advantage of the situation that's there. So most of the time we think of biofilms as being bad, so we mostly hear about the bad ones, right? So if you didn't know, the plaque on your teeth is considered a biofilm. Other things that you hear about are the ones that are, in the medical fields, where you hear about things that are contaminating like surgery instruments, or the lungs of cystic fibrosis patients, things like that. Those are all like the bad types of biofilms that we hear about. But there are actually some good biofilms. So there are biofilms in soil that actually contribute to filtering out groundwater. So, but in any case, these are all situations where you have these communities of bacteria, and then they can be single species or multiple species, but they are now kind of attaching to a surface and growing there. Okay, so again, they form when nutrients are plentiful a lot, and so this whole process of actually creating a biofilm is going to be cued by certain environmental signals, right? So the bacteria need to know that, oh yeah, this is that all of a sudden there's tons of nutrients here. This is a good place for me to kind of hunker down. But then also they are relying on each other to talk to each other, right? Because they're trying to form this community together. And so they do that through this mechanism called quorum sensing. So they use different chemicals to basically be able to talk to each other and figure out how much of your friends are actually already over there. So it's a way for them to kind of measure cell density. All right, so how do biofilms develop? So it goes through this whole cycle. I think I already posted the video on Canvas. If not, I will later today. But basically, so there's some kind of signal, right, that the bacteria now think that this is a good environment for it to try to stick to. So you have these different bacteria that use their flagella to kind of swim to a surface, and then they will start attaching to that surface. Once they attach, they will actually lose their flagella, and then they start forming these microcolonies. So you see in here in the microcolonies, you kind of see this shaded regions here on the pictures. They're starting to kind of coat their surface with polysaccharides or glycoproteins. And then now, again, they're using those quorum sensing signals to kind of tell each other, yeah, we found a good spot, come and join us over here. And they're trying to detect how many they're actually forming, because if there's too many of them, right, they're just going to use up the nutrients too fast. So it's a balance. But eventually, once they start getting to a high enough population, they're going to form these exopolysaccharides. So these are essentially going to coat and protect the bacteria from things that are essentially going to try and break it apart. So these are exopolysaccharides. These are the things that contribute to antibiotic resistance. So biofilms are harder to kill by antibiotics because of this extra coat here. And so eventually, they film these, what their book is calling them, biofilm towers, and they're just happy in there with all these nutrients. They're protected from the environment. But eventually the nutrients may run out, right? And so they will start to actually dissolve and break apart the EPS layer, and then they're going to kind of send out a few bacteria at a time to kind of go look for the new best place to go ahead and live. And eventually, if this runs out, the whole thing will kind of dissolve. Any questions about this? Yeah?
What's the main purpose of forming a biofilm?
It's mostly just they've realized that there's a good nutrient source there, and then also it's protection for them, because when they get into this formation, it's harder for other things to come and kill it and disturb them. All right, so that was a situation of what cells do when there are high nutrients, okay? So now in the case where all of a sudden there is a low nutrient environment, they're now in the place where they need to find other sources of nutrients, or they need to figure out, like they certainly just can't sustain logarithmic growth, right? They can't just sustain doubling according to their generation time. They need to figure out something else that they can do. So we can consider that something like environmental stress. But they essentially can go, and this is species specific, so different species will do different things. And so we'll just go through 3 different examples. But they will go through some reprogramming to form these different cell structures to help them survive. So one example is are these fruiting bodies. So this is from the species Myxococcus xanthus. So essentially they will form these clumps. They will aggregate into like 100,000. So this is something, on the order of 100,000 of these bacteria that are coming together and forming this cell structure, this fruiting body structure. So what this allows is it creates an environment for spores to form, and then those spores can get transported, and they will get dispersed, and they will hopefully try to figure out a new place to go and start living there. In the case of these heterocysts, so this one's a little bit different. This one essentially, it allows cyanobacteria to take advantage of kind of 2 different ways of getting energy and things. And we'll talk more about this later in the class. But essentially, these heterocysts allow the cyanobacteria to fix nitrogen. Something that we're going to learn about later is the enzyme that actually fixes nitrogen is very much inhibited by oxygen. But the cyanobacteria are also performing photosynthesis using oxygen, so they figured out a way to develop these structures for them to be able to do both and get energy in 2 different ways. All right, so the last example that we'll talk about are endospores. We've brought this up a little bit when we were talking about the experiments to disprove spontaneous generation. But so these endospores, they are formed by certain gram-positive bacteria, so Clostridium and Bacillus. So things that you've heard of probably, like tetanus, botulism, anthrax, those are all kinds of things that form these spores. The key thing about them is they're heat and desiccant resistant, so they're essentially very hard to kill, and they can survive for a really, really long time. So a lot of these kinds of species are soil bacteria, and sometimes you can find that these spores have survived 50 to 100 years. So the main idea, again, this whole process is triggered by starvation, so when nutrients are low, right? So you don't have to know all these steps in the process, just the big picture. The idea is that while it's, instead of going through replication and dividing into 2 cells, what it's actually doing, it's starting to form this spore, right? And so the spore, the key thing about the spore, again, it's similar to the biofilm in the sense that it creates this extra protective layer, right? So this protective layer is the thing that is going to protect it from different heats, the heat and different chemicals. Okay, so this is instead of the replication. And so we heard of terms like vegetative cells, right? So that's when the cells are actually growing and doubling normally. That's versus the spore, these spores that form in the cells, that they're actually not replicating, they're not doing anything, they're just kind of surviving until conditions are better.
Is it certain gram-positive bacteria? Is it gram-negative? You're not doing the spores?
No, not this type of spores. All right. Okay, so now so that was it from the rest of last class's slides. So today we're going to move on. So we talked about the growth cycles and how the bacteria are actually growing. So now we're going to talk about what things are actually influencing their growth. Okay. And then at the end, we'll talk about how they actually also die. All right, so microbes, they have both the fastest and the slowest growth rates of known organisms, right? So I've been telling you that E. coli doubles at about 30 minutes, right? So but there are also microbes that grow in these hot springs that are doubling at 10 minutes, so much faster than that. And then on the other end of the spectrum, things that live in deep sea sediments, it might actually take them 100 years to actually double. So that you can get the full spectrum, and again, it's going to depend on the environmental factors, right? So what do you guys think determines the differences in the growth rates? What kind of things can affect growth? Yeah. Yep. Somebody else had something. Anybody else? Yeah.
Available nutrients.
Yep. I'll write some of these down. Somebody else had their hand up over here. Maybe not. Somebody else.
Like pressure.
Pressure? Yep. Yep.
The concentration of light.
Okay, so yep, so light, if there are species that depend on light, it could be. Yeah.
Oxygen.
Yep, for sure, oxygen. Anything else?
Osmolarity.
Yep. Osmolarity. All right, I think this is essentially the major components, right, that are going to affect growth. And so what we're going to go through and talk through how all these different things affect the growth of microbes. And then, so here are some of the examples. So we hit all of the main ones, right? We were also, what's not on this list is nutrients, but we were just talking about nutrients in last class. So the kind of theme for today's class is we're going to be talking about extremophiles, right? So extreme meaning anything outside of normal growth conditions. And normal growth conditions, because we define them, are essentially the conditions that we live in. And so, for pressure, things are at sea level, temperature 20 to 40 C, near neutral pH. This is actually a very relatively low salt concentration. And then we're assuming in these cases that the cells have ample nutrients, okay? And so anything outside of this will be considered an extremophile, and you can see some of the names in here, but we're going to go through based on the different categories, they have different extremophile names. All right, so the first thing we're going to go through is temperature. So the first thing that you need to understand is that microbes don't actually control their temperature, right? They are just being exposed to a temperature, and they have to learn how to deal with that. Okay, so and based on the temperature, right, the different species, they all have these optimum growth temperature. So why do you think that? So what do you think is the major thing that temperature is affecting, or some of the major components that temperature is affecting in a cell? Yeah.
Protein.
Yep, how so?
Well, depending on the type of protein, it could kind of denature it.
Yep.
Denaturization and kind of affect the shape of the protein itself.
Yep. Saw some other hand over here somewhere. Yeah.
Enzyme activity.
Yep. Why do you say enzyme activity?
Because some enzymes perform at certain temperatures, and so they're better able to perform at these certain temperatures compared to other temperatures.
Okay.
So they're more efficient at these temperatures.
Yep, efficient is an important thing when we talk about temperatures. Is there something else? No. Anybody else? Yeah.
Maybe like if it's a warmer temperature, the gases move more and at a higher rate, so
Yep.
They're available at a higher rate.
Yep, so one of the key things that at temperature essentially is if you think of your chemistry, everything is moving faster, right? Molecules are moving faster, the bonds are going to be vibrating faster, right? Did you have something else?
Oh, I was just going to say that the temperature affects the membrane.
Yep, exactly. We already talked about how temperature affects membrane fluidity, right? So that is another key thing in terms of temperature. All right, so the other things that it can affect, right, it can also, so in addition to proteins, it can also affect DNA and RNA stability, right? If just anything essentially that it has a chemical bond in it, the temperature is going to affect that, right? Because essentially it's increasing molecular motion. And so all of these different molecules, they have this optimum temperature that they are going to actually function best at, and it's going to be dependent on what they're made of. So the specific amino acids, the specific nucleotides, that's all going to affect at what temperature is their optimum. But in general, you'll achieve the fastest growth rate when the temperature matches essentially the efficiency temperature of all your proteins. All right, so growth rate, so we talked about this is like, I told you it was proportional to the inverse of generation time, right? So this is why I told you it was proportional, because it essentially goes by something similar to the Arrhenius equation. So you've probably learned this in chemistry, right? I don't expect you to know the equation. Yeah, go ahead. Okay. So this line here is an Arrhenius equation plot, okay? And so what you can see here is that there is this region here where the growth rate follows this very closely. So what's essentially happening in this, so for this bacterial species, right, it's doubling, the growth rate is doubling for every 10°C rise in temperature. But you see that, so this blue line here is growth of this bacterial species at these different temperatures. This is like calculated experimental growth, right? But you see at the higher temperatures here and then at the lower temperatures here, they don't actually obey the Arrhenius equation. So what's happening at the higher temperatures, the growth rates don't follow this essentially because things like proteins are denaturing, right? The DNA and the RNA, they're also starting to denature. And so you end up having a slower growth rate because things are not functioning properly. And then when you get too cold, mostly it's kind of the opposite direction, right? So everything's moving a lot slower. Things just slow down so much, so that's why things that's how you end up getting those things where like the 100-year kind of doubling time, because they are kind of going, but they're just going really, really slowly. And so again, it doesn't obey this law. All right, so we can classify microbes based on their optimum growth temperature. And so that's what these different file names are referring to temperature. So the extremophiles in this case are the extreme thermophiles, the thermophiles, and the psychrophiles. The mesophiles are the kind of the normal condition bacteria. So the ones like our lab strain, E. coli, and things like that are the mesophiles. So you can see at this, so what is this plot telling us? So it has generations per hour, right? So essentially how well these bacteria are growing, and it has this range of temperatures here on the bottom. So you see that the typical growth range for these different categories is between 30 to 40 C. So some of them are smaller, so like the psychophiles are smaller, but these ranges here, right, about 30 to 40 C for the different classifications. But each one of them has this kind of optimum temperature, right? So so the peak of each one of these has this optimum temperature, and so that's for these classes of microbes that their proteins are working most efficiently is here. All right, so what are some strategies that these different types of bacteria use to actually survive in these temperatures? So we'll start with the psychrophiles, so the ones that grow in the very, very cold temperatures. In these cases, you'll find that the protein, so general categories of things, right? The makeup of the protein, so like which amino acids are actually making up the protein structures. So these proteins, for the psychrophiles, they're generally more flexible, because if you imagine it's trying to combat freezing, right? We already talked about how the membranes are more fluid, but they also have things like these antifreeze proteins, and they have other cryoprotectants to essentially, again, right, they're at the temperatures of their environment, and so if they have all these other cryoprotectants and proteins to help them essentially change the freezing point for themselves so that they don't actually freeze when, you know, it's 5 degrees Celsius outside.
Energy to function. Does that mean they're like less complex, or are they more complex?
It's hard to say. It's hard to grade them on complexity. Energy just meaning it doesn't require as much like molecular motion for them to actually survive and divide. Does that make sense? Yeah. If you were to ask me about complexity, I would think that they're more complex than, you know, your standard mesophile that survives at these, you know, standard conditions, but that's just kind of opinion, right? On the other extreme, so the thermophiles and the hyperthermophiles, now we're thinking about the opposite direction, right? So these enzymes, they have to be more stable, so because you don't want them to denature. So how they can do that is they can actually change, again, like I said, the amino acid composition. So you'll find that there are less glycines in the proteins for extremophiles. And again, they're going to have, yep.
I have a question about the psychophiles.
Sure.
So it says that they were.
Okay, so again, yeah.
Any specific amino acids for psychophiles?
No, no.
Only glycine?
Yeah. So for the thermophiles, so again, they have other proteins that are going to help them out. So in this case, they have the chaperones and DNA binding proteins that are essentially preventing denaturation and then helping them stabilize the DNA so that they're not going to melt at these higher temperatures. Okay, and then we also, again, the things that we had talked about before, the membranes, the membrane compositions to make them more rigid. All right, so now we'll move on to pressure. So the name of the extremophiles when regarding pressure are called barophiles, or these piezophiles. So again, these are microbes that have adapted to be able to grow at really high pressures. So if you think about the sea level being 1 atmosphere, we're talking about 1,000 times that, right? And so the other now we'll just go through the terminology. So there are the barophiles. These actually require higher pressures to survive. And then there are some barotolerant microbes that they can, they're happy to grow at atmospheric pressure, but they can actually tolerate the higher pressures, although they will still die at these the higher pressures. It's a little bit easier to see on this plot. So this plot, again, we're looking at growth rate, and this is increasing pressure, right? So this black line here is your kind of your standard microbe that can only survive at the standard pressures around here. And so as you increase the pressure, it's not going to be able to grow and survive there. The red line here, so these are the ones that I was just talking about, the barotolerant ones. So these they can tolerate, so they're happy to grow here at atmospheric, and then they can tolerate pressures, increasing pressures up to a point, and then they actually, then they can't survive anymore. So now again, this is versus the barophiles, right? These are actually require the high temperatures. You'll see that they don't actually grow, right, at the atmospheric conditions. All right, so many of the barophiles, they're actually also combined psychrophiles or hypothermophiles, because essentially in order to create these really high pressures, they're also in these different conditions where it's like really cold, right? So these ones, we actually know much less about how they actually survive, right? So we had come up with some strategies for how different microbes survive the different temperatures. We don't know as much about the pressure, but we do, we can imagine, right, that many of them are going to have increased membrane fluidity because a lot of them are growing where it's really, really cold. And then they're going to have some way to have specially designed membranes and then protein structures, right? Because they're going to be able to have to survive these really, really high pressures. So just for reference, right, E. coli ribosomes, they dissociate above 60 megapascals. So here, right? So essentially, if your ribosome's dissociated, you can't be translating any more proteins, and so the microbes that are surviving out here, there has to be some kind of strategy for them to be able to survive there. We just haven't figured that out yet. All right, so the next thing that we're interested in is osmolarity. So hopefully you know a little bit about osmolarity from your chemistry classes, but osmolarity is just essentially this measure of the number of solute molecules in a solution. Okay, so what matters to us is what kind of place that the actual microbe, the environment that it's in. So if you have an isotonic solution, so where essentially the solute concentration is equal on the outside of the cell and on the inside of the cell. Or you can have a hypertonic solution. So in this case, right, the solute, so the red dots, right, these are the solutes. The solute concentration is higher on the outside, so in the environment that it's living in, versus the hypotonic solution where the solute concentration is higher on the inside of the cell. All right, so why does this matter, right? So in general, right, this is just naturally how the world works when it encounters a situation like this where you have some kind of semi-permeable membrane. So the lipid bilayer, remember, is a semi-permeable membrane that water can pass through, right? So water is able to pass through the membrane, but it does so pretty slowly, but it will do it. So what it's going to want to do is reach some kind of equilibrium, right? So it always wants to kind of hit this isotonic state, and so it's going to move water either out of the cell or into the cell, and essentially the effect that can have is what you see in these pictures here, right? So where things are now essentially like shrinking the cell down, and then in the other case, it's going to be increasing, and so it's going to create more internal pressure, which now the cell is in danger of actually lysing, because if you keep adding more and more water into it, it's just like a water balloon, right? So you fill up the water balloon and keep going. Eventually, there's just too much pressure and it's going to pop, right? So same thing with the cells. All right, so some of the strategies and what are the things that the cells actually do to deal with the different osmolarity situations that they might be in. So again, like I said, the water can pass through the membrane, but it does so pretty slowly. And so the cells have these aquaporins. So same thing that we were talking about before, membrane proteins that help transport different molecules. This one is for water, and so this essentially allows the water to move across the membrane much faster, and so this can help protect them from some of the osmotic stress. So strategies for microbes that are in the different kind of situations. So if they're in a hypertonic situation, right? So there's too much solute on the outside. It always wants to be balanced, right? So instead of trying to essentially deal with the water change, what you can also just, the other strategy is to just increase the concentration of the solute inside the cell, right? So it wants to protect water from leaving so that it doesn't shrink up like we saw in that picture. So things, can make, so these things like proline, glutamic acid, potassium, and betaine, these are all what we consider compatible solutes. And so it can start actually generating these things or importing more of them, using their membrane proteins to let more of these things in. And so that way it doesn't have to lose the water. And then in the opposite case, so for the hypotonic solution, right? So now you have you have too much on the inside, so you want to let some out. They have actually these pressure sensitive channels. So again, it's the same idea, these membrane proteins, right, that are allowing things in and out. These are going to actually be triggered by pressure. So if your internal pressure is getting too high, it can actually leak out some of the solutes, so that way it's not dealing with the pressure caused by the water change. All right, so what are the extremophiles that relate to osmolarity? So we have the halophiles. So these ones are another one. So these ones these ones are now requiring high salt concentrations to grow. So they grow in these kind of 2 to 4 molar areas. So this is 20 to 30% salt, which is compared to, if you think about actual just seawater, is only 3.5% salt. So that is a huge difference, right? And so these microbes, again, are requiring high salt. So you couldn't put these in like a low salt condition. They wouldn't necessarily survive. But they've, again, developed these different methods to kind of deal with the high salt. So they use special ion pumps to secrete sodium, and then they replace that with other cations. So they have different mechanisms to be able to deal with the high salt concentrations. All right, so the next category is pH. So if you remember now, pH is just a measure of H+ ions, right? So in the case of in biological activity, we're thinking about hydronium ions, right? And so here are some examples of like the pHs of different common things that you might know of. So what do you think, how does pH, so H+ concentration, actually affect enzyme activity?
Well, it could denature a protein or an enzyme, or it can chemically increase in H ions will bind to other, ions on the enzyme that are necessary for interactions.
Yep.
So it takes away its function.
Okay. Yep. Any other thoughts on how H+ ions can affect enzyme activity? Yeah. Yep, why?
Because of the charge.
Yep.
What'd you say?
The enzymes might go through conformational changes because of the different charges. Anybody else? All right, so if you just remember, right, the amino acids, they're made up of all these chemical groups that are essentially have charges to them, right? Like the amino groups, the carboxyl groups, some of the side chains, right? They have their acids, right? They have charges to them. So the H+ concentration is essentially going to affect the protonation of these groups, right? And so when that changes, like we've had just talked about, that can actually change the conformation, so the shape that the proteins actually fold into, and that shape, most of the time, right, has everything to do with how these proteins function, right? So even if it doesn't change let's say just, the H plus concentration, let's say it doesn't necessarily change the shape of the protein, it can still change the catalytic site, right, of an enzyme. So if it's required for a charge to be there to actually do the enzymatic activity, that charge might actually change depending on the pH that it's in. All right, so the majority of enzymes, they operate best in this range, so pH 5 to pH 8.5. But as you can see, right there, and as you can imagine, there are many different pHs of different solutions that you could expect that bacteria to live in. Yeah.
I have a question about the way it works. So at the lower pH is less hydrogen molecules in the solution. Is that right?
So the lower pH, so this is the concentrations here, and so the lower pH has more hydrogen.
Oh, it has more
Yeah.
hydrogen?
So it's opposite because you're taking the logs.
Okay, more hydrogen. So then the higher hydrogen causes the hydrogen in I guess in the structures to leave and make it more. You know, like how the carboxylic acid.
Yeah. So instead of having an O-, they would now all be protonated. So the H's would be attached to them if there was in a higher concentration of hydrogens.
So the carboxylic acid, the more H's would attach to it, not leave, right?
Yep.
Okay.
Yep. Any other questions? All right. So before we move on to that, so the major thing, one of the bigger things to understand in the case of pH, right? So now the microbes, they actually have the opportunity. They're not actually just succumbing to the pH of the environment, right? So in the case of temperature and pressure, like they don't have a choice. They don't have a way to actually control that. But in the case of pH, they actually do. And so these are the different kind of categories according to the pH. There's a typo in this. So this should actually be 14. We'll just go through the categories first. So the neutralophiles, so these are the neutral-ish pHs, right? The acidophiles, the things that are growing in more acidic conditions. The alkophiles are growing in the more basic conditions. But there are all these different strategies that they use, right? So in the case of the neutralophiles, like I said, they know what their optimum pH is, but they can actually adjust the metabolism that they're doing, so whether they're producing more acidic or more basic compounds to kind of counteract what's happening in the environment. For acidophiles, they have these altered membrane lipids with the high levels of the tetraether lipids, so this actually decreases proton permeability. And then for the alkalophiles, they have cell surface barriers that protect their internal proteins. And one of the things that they also adapt to now instead, so microbes that are living in these high pH environments, they can adapt to using a sodium motive force instead of a proton motive force, because they're going to have much more of the sodium ions available to them than they would the hydrogen. So again, the difference with the pH, it's important to remember that the intracellular pH is not necessarily what's on the outside, because during metabolism, the cells are doing so many different things, and it's actually changing the pH inside of the cell. All right, so the last thing that we need to consider is oxygen. So oxygen, we actually consider oxygen as something extreme, right? Because to you that might sound weird, because we need oxygen to survive, but to the microbes, when oxygen breaks down, it actually forms these reactive oxygen species. And so things like hydrogen peroxide and superoxide radicals. So these things are very harmful to the normal function of the cells. It can damage the DNA in your cells, and that's going to be very bad, right? You can imagine that the cells wouldn't want that, and that's going to be really hard to survive. So in order to actually survive this, you can't stop this from happening. You can't essentially stop hydrogen peroxide and these superoxide radicals from forming. So what they've had to do is they've had to evolve these methods to essentially deal with them and make it so that those are no longer harmful to them, so that they can survive in oxygen. But so why would they need to survive? Why would they want to survive in oxygen anyway? Well, we'll learn later when we talk about metabolism, and maybe you've learned in your other classes, that oxygen is great when they're trying to make energy, right, as a terminal electron acceptor. So that's one of the best sources that you can have. And so there is a benefit for them to be able to live in oxygen. But they've had to come up with these different methods to be able to actually survive doing so. And so we categorize the different microbes based on whether or not they can survive in oxygen, whether they need it, whether they don't. And so these are different categories. So strict aerobes, so these ones only grow in oxygen, so they can't grow without it. This is versus the microaerophiles. So these ones, they still need oxygen to grow, but they can only tolerate very little oxygen. So the oxygen level is much lower than the strict aerobes. And so you can imagine, so essentially what's happening for the microaerophiles is they have less of those enzymes that are dealing with the reactive oxygen species. Okay, so now we have the strict anaerobes. So these ones, they do not have these safety mechanisms, so any sign of oxygen and they're gonna die. The facultative anaerobes. So this one has always been tricky because it's facultative, but then the word is anaerobe. So, but these ones can actually live in oxygen. So that's the tricky part of this one. So they can live with or without oxygen. So E. coli is a really good example of a facultative anaerobe, because we grow that in the lab with oxygen, but it also grows in your gut where there's no oxygen. And then the final category are the aerotolerant anaerobes. So they grow in oxygen, but they actually use anaerobic metabolism. So they won't use oxygen as their terminal electron acceptor. All right, so we've been talking about how microbes actually grow in all these situations, but how do they die? How can we actually kill them? So this is just some terminology, and they have these fine differences between them. But essentially, so sterilization is when you essentially kill everything, okay? Because some of these other ones, so like disinfection, so this is only killing or removal of pathogens from inanimate objects. So the term pathogens is what matters for disinfection. There could be some like harmless bacteria or whatever on that surface, but so those that's not what we're concerned with when we talk about disinfection. Antisepsis, so this is removal of pathogens from surface of living tissues. And then sanitation, so again, this is only reducing microbial populations to safe levels, so it's not killing everything. All right, so just like we had cell growth and we had equations and ways to model cell growth, we can also model cell death. So the D value, so this is decimal reduction time, is essentially the length of time it takes for whatever agent that you're using to kill it to reduce the population to 90%. And so that's what you can see by this plot here. So if this is like 100% of the bacteria that you had, now this is a log plot, so this is now 90% have been killed, so how long that time actually actually takes for you to do that. And you can measure that this is dependent on whatever antimicrobial that you're using. So some of the things that we can use to actually kill microbes, so temperature, combinations of temperature and pressure, irradiation, filtration. So we kind of talked about filtration when we were talking about the discovery of viruses. They knew that you could capture the microbes with these different size filters. So that's what filtration is referring to. We have chemical agents and even some biological agents to kill. I am not going to have enough time to finish this, so we'll stop here.